Hi, let's discuss about the antibody structure and classes. And we want to discuss about the functions of antibody for a protection from antigens, which is non-self or foreign. So what are the antibodies? Antibody is a specialized protein. You remember proteins that can inactivate pathogens directly or mark them for destruction by other immune elements. So antibody is a very interesting protein and very important. And that looks like a Y shape. Let me show you its structure. So antibody looks like a Y shape, and that tip of this finger is where antigen is binding site. So you can consider it as the antigen as a foreign, which is recognizable by antibody, and the relationship for this connection is like key and lock structure. So look at this. So what is this antigen? So antigen is a substance that can be recognized by antibody or immune cell receptor. So you can geometrically consider this antigen like this. Let me show you more general way of this antigen, which is a yellow. You know, the pathogens or anything that has a shape which is not non-self or foreign. So we have our own antibody, very diverse antibody system, which specifically fits into this special shape of this antigen. So we should have lots of antibodies which do not necessarily recognize our own molecule, but the foreign molecule. And this even one antigen, you can see there could be a lot different kind of shapes and positions which is recognizable. So that we call the part of antigen that's recognized by our immune system is called epitope. Epi means a surface, so it's on the surface, and this is determining. So the part of antigen molecule to which an antibody attaches, that is also called antigenic determinants, okay? Because we are determining this antigen by our antibody. So let's go through a little bit of the definitions of antigens and antibodies. So antigen is a target to which an antibody binds. And what can become antigen? It can be a protein from foreign molecule or polysaccharide or small molecule. And antigen thus can be associated with external bacteria or virus, or even, let's say I need a kidney transplant from another person, that transplanted organ is now, you know, it's a foreign, so it can be considered as an antigen. Or whatever entity that is foreign to the host is called antigen. So that specific molecule which our immune system produces is an antibody, and the shape is related to a kind of globular protein. So that's why we call antibody, in other words, called immunoglobulins, or simply write the IG. So the reason is antibodies are globular plasma protein, which is produced in response to and counteracting a specific antigen. You see, I put a specific. Specificity is a very, very important characteristic of antibody. So when an antigen enters our host, our body, that antigen will stimulate our immune system, often leading to the production of specific antibody molecule, which can bind to that specific antigen. So which is done by the antibody production is done by a plasma cells or antibody producing cell. And that plasma cell is matured from B cells. And you can wonder, where comes the B from? The origin of the B is from bursa, which is from a bird's. We found out that specific sect, there's a lot of B cells. And the B cells are originated from bone marrow. So that's how I remember. So bone marrow is where B cells originate, a kind of circulating white blood cell is a. So that antibody, if the antibody is produced by a single clone of cell or cell line, then the antibody will be completely identical. And that identical antibody we call as a monoclonal antibodies, or simply MABs. So MABs should bind to the same epitope. So because it's totally identical antibody molecule. So let's get into a little more specific into the structure of antibody. So antibody should have a site which recognize antigen and binding. So it has a pocket. So typical antibody has two arms so that it has a bivalence, okay? So it looks like this. So it's a Y shape. You can see there's like 4 chains, 2 are short. So we, in terms of mass, we call light chain, 2 are long, which is heavy chain. And you see that binding sites are the 2 tips. So that's we call antigen binding site, which is composed of both light and heavy chain. And that part has to be a very, very diverse to figure out all the invading outside of the world. So it has a huge variability. We call hyper variable, okay? So that's a variable region, while the rest do not have to be different. So the rest should be constant. So we call it a constant region. All right, so that variable region of each chain should contain hypervariable region, or we call CDR, or because it attaches to antigen like a complementary match. So we call it a complementary determining regions, or CDRs. So these CDR form a 3-dimensional structure to bind to specific antigen. Okay, so this Y shape is just a conceptual ways, but the reality looks a little more complicated. So you can see this tip of this region, this 2 part, and then the body part. And so for study this, we have to chop out so we can see it as a fragment. So we name it as this constant region as we use F, stands for fragment. So there are 2 fragments. One should be that antigen binding site. So we put it as FAB, meaning antigen binding site fragment, while the rest constant as a constant fragment region, or FC region. So you can see that this light chain is about 24 kilodalton, and the heavy chains are about 50 kilodalton. So both you combine this, total about 70 to 80, and there are 2. So we have antibody like this has about 150 kilodalton, which is a quite big macromolecule. And you can see here, this blue is a heavy chain, and the red is light chain. And you can see this yellow is also a heavy chain. You see this variable region, so V from the light chain, V from the heavy chain. And the C stands for the constant region from light chain and heavy. Constant region, this H stands for hinge because it's kind of flexible. It has flexibility.